tissue level of organization. Now, if you recall back in our levels of organization in biology, in terms of life, we look at the cell being the smallest unit capable of sustaining life. Humans, of course, are going to have millions upon millions of cells. So when you put different types of cells together, you get types of tissues, and that's what we're going to be discussing in today's lesson, the different tissues of the body. Now, this is going to give a brief overview of the 4 major types of tissues that we are going to be covering throughout this chapter. But again, let's look at the cells first. So talking about the cells, we can see that the cells are that basic unit. You have so many different types of cells in your body, each with a very specialized purpose. Those cells are going to secrete different things to the outside of their body. That's why you have these extracellular materials and fluids. So each individual cell can make certain things and then can actually export them out of the cell to create extracellular material and fluid. We will oftentimes refer to that as the matrix that's going to be around it. So cells are going to be bathed in a fluid and have different proteins and substances on the outside of the cell. When you have those cells working together collectively with all that extracellular material and fluids, they're going to form these different tissues of the body. So like I mentioned, there are 4 different tissues of the body. We will be covering epithelial tissue, connective, muscle, and nervous tissue. The first segment, we're going to start with epithelial tissue. Now, when you have these different types of tissues coming together and working together, then they will form organs. So organs are going to have specialized functions that they're going to do within your body. And then if you take organs that are collectively working together, you can create organ systems. So when we talk about your respiratory system, your cardiovascular system, those are all organs who are working together and interacting to do a big purpose within the body itself. And then, of course, when you put all those different organ systems together, you will get the whole organism. So let's dive into epithelial tissue first. In terms of function, the epithelial tissue's major function that we're going to look at is physical protection. That's because epithelial tissue is going to be creating linings of the body. So the outside of your body is lined in epithelial tissue. The outer covering of the organs are all lined in epithelial tissue. And not just the outside of it. The inside is also going to have linings. So when you have ducts or tubes that are running through your different organs, all of those tubes are going to be lined with epithelial tissue as well. So because epithelial tissue is going to be a covering for so many things, another major function it's going to have is to control permeability. It's going to determine what's going to be allowed to enter and exit that particular organ, so it's going to control and regulate those items. Another big part is it's going to provide sensation, specifically when you think of like the skin. So the outer portion of it, your skin, is going to have that epithelial tissue that's lining it. Embedded within that epithelial tissue, you will have sensory receptors. Those receptors are going to receive signals so that you know what's going on the outside world and whatever's making contact with the epithelial tissue. And another function we're going to look at is that it's going to produce specialized secretions. That's because when we have glands in your body, those glands are associated with your epithelial tissue. And glands will create these secretions that are needed for different aspects of what your body is going to be doing. But those glands are going to be derived from the epithelial tissue. Epithelial tissue, compared to the other 3 types of tissue that we're going to look at, has very specific characteristics that we look at. So you can see the 5 that are listed there. So let's hit polarity first. Polarity is referring to that fact that epithelial tissue has 2 distinctive ends to it. So we have an apical or top surface and then a basal surface that's going to be at the bottom. The basal surface is physically attached to another tissue that could be muscle underneath, it could be bone underneath, some other sort of tissue is where the basal surface is going to attach to it. The apical surface, on the other hand, is going to be a free edge. So if you're thinking in terms of your skin, it's literally the part you're touching on your skin where it's open to the outside environment. Now, specifically, when we are looking at polarity, oftentimes with the apical surface, if it's an internal structure, we're generally going to have structures on that apical surface that are going to help with whatever process is happening. So you can see on these 2 different cells, this one is showing microvilli structures, and then this cell over here is showing cilia. So cilia and microvilli structures are oftentimes found on that apical surface, which will allow us to do whatever function for that particular tissue. So for instance, when we have cilia are basically little hairs that beat together cohesively, and so they can push things to move in a certain direction. So if you were thinking about like your throat and you have mucus that's being moved around, the cilia will beat in a way to move the mucus into a certain direction. So that's one function that you will find, because this is a free edge, and that free edge is open to where the mucus would be coming in and out. You can also have microvilli. Microvilli are little finger-like projections. They don't beat back and forth to move things, but they do allow us to greatly increase our surface area. So you will find these in places where you want to have rapid diffusion across the surface. Because remember, epithelial tissue is also going to help to regulate what's going in and out of the cell. Well, if you want to absorb nutrients, you want to have as much surface area as possible, because greater surface area means you're going to have a greater rate of diffusion, and then you can bring things like nutrients in very quickly. If we're thinking about a tubule that's being lined by epithelial tissue, that tubule, which allows things to flow through it, that apical surface is where you're going to have the friction with whatever is flowing against that particular surface. It is also going to exhibit cellularity, which means that you will have different types of cells that are going to be fused or attached together to create this tissue. So when we look at this little figure that we have right in here, we can see that you have, of course, the basal surface, which is attached to something underneath another tissue here, but then you also have these lateral surfaces. The lateral surfaces are where going to be the individual cells within the epithelium are also attached together. So they're not like freely floating and can shift away from each other. They are actually going to be attached together. And again, that's what we're going to look at with this attachment site too. So attachment to some sort of tissue that's going to be underneath it. You're going to have a non-cellular layer that's going to be underneath. That's this basement membrane. So this basement membrane is formed to act as an attachment point between the epithelial tissue here and generally connective tissue that's going to be right below it. The cells are going to be creating that extracellular matrix. So they're going to be creating proteins and fluids and things that will allow them to cross this junction to create this basement membrane that allows it to attach to the underlying tissue. Epithelial tissue also exhibits avascularity. Now, when you see A in the front as a prefix, it usually means without. So we are without vascularity. Vascular means referring to blood vessels. So epithelial tissue has no direct blood supply. You do not have blood vessels, arteries, and veins that are running through the epithelial tissue. That means in order to get nutrients are going to have to diffuse from blood vessels that are going to be in the underlying tissue, like the dermis that's going to be under there, or some sort of connective tissue. It's also going to exhibit regeneration. Now, cells in your body in general are able to regenerate. They're going to go through mitosis to continue to create more cells, just like the ones that you had. But because the epithelial tissue tends to have a lot more friction with the outside environment, if you're talking about the skin, or even the lining on the inside of tubules, things are flowing against them. And so because there's a large amount of friction, the regeneration that you have within the epithelial tissue is generally a lot faster than you will have for other cells. So the turnover rate that we have for epithelial cells is going to be a lot higher. They're going to continually replace because they are going to have stem cells that are associated with it, and those stem cells are going to be able to divide and then replace the different epithelial cells that are in there, so you can have that regeneration. Now, we mentioned that cellularity, and with that cellularity, the cells are going to be attached to each other at those lateral surfaces. So when you look at the lateral surface where one cell is adjacent to another cell, they are going to actually be connected to each other, because those cells are creating what we refer to as CAMs, or cell adhesion molecules. The cell adhesion molecules are going to be part of that basolateral surfaces. So the lateral surfaces and the basal surfaces are going to create these proteins, generally proteoglycans, that are going to extend from one cell to the other in that extracellular matrix, and they're going to hold them in place so that the cells are not going to separate from each other. We do also have specialized cell junctions, though. So these are going to be specialized connections that we have that allow the cells to do different functions. So we're going to first talk about gap junctions. So looking at gap junctions, that's going to be letter B right here. So we're looking at a close-up between cell here and then this cell over there. So for this particular cell, what we're looking at with a gap junction, we are going to create what's referred to as a connexons. These connexons are proteins, peripheral proteins that extend from one cell over here, and then they attach to another protein that's produced by this other cell. And then they basically create a little tunnel or a gap between the two cells. This is for communication purposes. This gives easy diffusion of certain small molecules that will be allowed to go back and forth between the two cells so that the cells can know what each cell is doing, and that's going to help with homeostasis. We also have tight junctions. So over here you can see in letter C, these are going to be tight junctions. So this is looking at this area up in here of the cell, and then you can see where they are going to have these tight junctions. So looking close up at this, the tight junction is going to be the fusion of two different outer layers. So it's almost like a zipper where you're sealing these two cell membranes together. That continuous adhesion that you have prevents fluids from flowing between the two cells. Now, we had mentioned before how there's like extracellular fluid. So all your tissues are bathed in this fluid, any living tissues that you have. Well, we don't have between these two tight junctions fluid that's going to be able to flow through them. Now, the other junctions, like this gap junction, there's a big opening over here between the two cells. So fluid goes between the two cells, as well as things going back and forth between the two cells here through the gap junctions. But we're talking about in between at that lateral surface that we're looking at. In a tight junction, you don't have fluid flowing between the two cells because the tight junction is going to seal that together so that they can't do that. This is going to make sure to control any kind of movement of solutes or fluids between cells so that it's not going to be able to go between the cells. It will have to actually be transported from one cell and then directly go to another cell from it. It can't just flow in between the two plasma membranes. The next we're going to look at are going to be the desmosomes. So here we can see a spot desmosome here. We have some hemidesmosomes over here too. So let's look more closely at this spot desmosome. So with the spot desmosome, here you can see some of those cell adhesion molecules, which are going to be anchoring and holding this cell to that cell. So the left and right cells are going to be anchored together. So the cells create kind of a sheet of cells within this tissue. And then you can see that you have these different filaments that are going to be in there that are part of the cytoskeleton that are going to help to tie these two together so that they're going to anchor them in place. This is going to allow us to resist stretching and separating the cells from each other. So the tissue is going to be flexible in that it can move, but the individual cells will not be pulled away from each other. Now, in biology and anatomy, we like to name things very particularly. So we classify things to group them in ways where we have names that make sense, or that's the goal, is to have names that make sense. So when we're looking at classifying our epithelia and putting them into different groups, there are two different characteristics we're going to look at. We're going to look at the individual cells and the shape of the individual cells that are making up that tissue. And then we're going to also look at the number of layers of those cells that are going to be present in order to name them. Now, when classifying the different types of epithelium, we can have simple epithelium or we can have stratified epithelium. Simple epithelium is just that. It's simple. It is a single layer of cells. When we talk about stratified, stratified is two or more layers of cells that are going to be in there. And then we have three basic cell shapes that we're going to look at. The first of which is going to be the squamous. Squamous are flat and scale-like cells. These tend to line cavities and are part of the mesothelium is usually what we end up calling them. But they have very thin and flat, and they have a very small nucleus that's going to be with them. The next shape is going to be cuboidal. Cuboidal are little cubes, or they're very short and box-like. They will have a large nucleus that's going to be within there, taking up a good majority of that cell space. And then our other category that we have are columnar cells. They are columns. They are very elongated and long. Generally, the nucleus within these cells are going to be oval in shape. So as you can see here, this is going to be simple columnar epithelium because we have just one layer. Oh, and look, we have cilia right at the top. Here we have stratified columnar. Here we have lots of scale-like flat cells, and that's going to be a stratified squamous epithelium. So we take into account both the cell shape and number of cell layers when we're naming it. And generally when we name it, we will say the number of cell layers first, then the shape of the cell, and then we always end it with epithelium.